Good morning and good afternoon, everyone. Welcome to join the webinar 3D Printing, a Game Changer for Medicine, the fifth installment of Connected series, which focuses on Sino-Swiss collaboration projects and research efforts. My name is Li Binggu. I'm the head of academic relations at Swissnex China, Science Consulate of Switzerland in China. We connect the dots between Switzerland and China in education, research, and innovation. Today, we are very delighted to have Dr. Ying Bao and Nivina Paunovic, both from Institute of Pharmaceutical Sciences at ETH Zurich with us, and share the latest development in their studies in 3D printing and its biomedical application. Now, a kind reminder to our audience, at the bottom of the Zoom interface, you can find the Q&A function. So please type in your questions during the webinar, and we will answer them in the Q&A session. Now, I would like to introduce our first speaker, Dr. Ying Bao, Group Leader at the Institute of Pharmaceutical Sciences at ETH Zurich. Dr. Bao received his bachelor degree in micromolecular materials and engineering from Hefei University of Technology and a PhD degree in polymer chemistry from University of Science and Technology of China. From 2012 to 2014, he completed a postdoctoral research at KU Leuven and became a Marie Curie Intra-European Fellow in the French National Center for Scientific Research at University of Paris Sud. Dr. Bao is currently leading an interdisciplinary project funded by Swiss National Science Foundation about 3D printed biodegradable personalized medical devices in collaboration with 3 different groups at ETH Zurich, Zurich Animal Hospital, and University Hospital Zurich. And now I would like to give the word to Dr. Bao. So Dr. Bao, the floor is yours.
Okay, thank you. I will share my screen. So can you see it? It's normal. Can you see the screen? Okay, great. Thank you. Thanks for the kind introduction of Li Bing. And today is really my great pleasure to talk about 3D printing with light. And I think most of the audience today more or less suffered from the COVID-19 during this quarantine time. You may already heard the story about this Italian startup company. They use the 3D printing to produce the valves for reanimation devices that saved a lot of lives of the COVID-19 patients in Italy. And besides these valves, 3D printing can also be used to print a lot of medical devices, like these testing devices for the coronavirus, or the personalized protective equipment, or the face masks, or even this 3D printing isolation wards. So 3D printing is quite useful in the fight against the COVID-19. Besides this, actually the first 3D printed splint has been used in surgery from 2013. And this 3D printed biodegradable device saved a lot of lives for these little babies with these native airway collapse. And the surgery has been successfully operated in the different university hospitals in the United States. And in 2016, we have the first 3D printed tablets approved by FDA then used for the epilepsy medication. And in nowadays, 3D printing has been widely used in produce the surgical models in the a lot of hospitals. Like this case in Mayo Clinic, the doctors can use the CT scan and 3D printing to produce both of the surgical models and also the implants. So before surgery, they can practice a lot based on the combination of the surgical models and the implants. So this can greatly increase the success rate of the surgery eventually. So besides the clinical application of 3D printing, the 3D printing technology is more extensively studied in the biomedical research. So for example, this work was published in Science last year. They used the 3D bioprinting to rebuild the components of the human heart, including this very complex multi-scale vascular structure. They also achieved the organ-scale 3D printing based on the native biomaterials. So this is a step forward toward the manufacturing of human-made organ. So 3D printing is so attractive and so useful. What exactly means that for 3D printing, or the more scientific word, additive manufacturing? And what's the difference between 3D printing or additive manufacturing with the traditional manufacturing methods? So for traditional manufacturing methods, you have this subtractive method and formative methods. For subtractive methods, normally you have a bulk material first. You need to remove the materials not necessary and finally get the object you want. In this process, you have a lot of materials that will be wasted and cannot be used anymore. For the formative method, you first have a mold. Then you inject your materials into this mold. After the materials get solidified, you take off the product. But for this process, you must have a very expensive mold first. Then need to be precisely manufactured, taking long time. In contrast, for the additive manufacturing, it's a rather straightforward method. We just use the concrete materials. We stack the materials layer by layer and design based on the CAD software. The whole process is quite straightforward and neat. For example, if we want to print a mug like this shape, we first need to design it with CAD software, and we convert into different slices like this. Then we send these files to the 3D printer. After the processing, we'll get this mug. So the whole process is very convenient and efficient. Maybe you ask the cost of 3D printing compared to traditional manufacturing. Of course, we can't print the consumables for everyday life using the 3D printing, because the traditional manufacturing can provide large amount of units with very short time. It's also very cheap. But for some specific area like the personalized medicine, we need 3D printing to design a specific shape or geometry. In this case, actually, the 3D printing is much cheaper than the traditional manufacturing, and also the time needed is much shorter. So actually, for the previous examples I mentioned about 3D printing are all based on this extrusion 3D printing. So which means the materials will be extruded from this nozzle, normally by heating or by shear force. And after solidified, the materials will form 3D objects. So for this method, the resolution of the printing is limited by the nozzle size. Normally, by a mechanical design, you can't get a very small nozzle for the printing, so the resolution normally is not so good. On the other hand, we have this light-based 3D printing method or the photopolarization-based 3D printing method. Normally, the materials will be cross-linked or photopolymerized to form the solidified products. And for this method, the resolution is only limited by the shape of the light. Normally, in this method, the resolution and surface quantity is much better compared to the extrusion-based 3D printing. So today, I will focus on the introduction of this light-based or photopolarization-based 3D printing methods. So the first example, or the most typical example, is the stereolithography, or SLA. I also entered the Chinese word for our audience from China. So if we want to talk about SLA, we must talk one person called Chuck Hull, because he actually coined this term stereolithography in his first patent and laid the groundwork for this technique. And he's also very successful in the business of 3D printing. But behind this technique, there's also another man called Alain Lemaitre. He actually filed another patent even earlier than Chuck Hull's patent. But he's not so well known as Chuck Hull because his French company abandoned his patent for business reasons. So afterwards, his career path was totally different with Chuck Hull. And if this is not the case, the history might be different, but this is life, c'est la vie, right? So for the materials for SLA printing, normally we call the materials resin. This is a liquid mixture in combination of different compounds. So the two major components are the photoinitiator and the photoactive monomers or macromonomers. So the photoinitiator can generate the active radicals under the light irradiation and cause the cross-linking or photopolarization of the monomers, and then form the solid products. Normally, we also need some additives to improve the 3D printability of the materials and also the resolution and the surface quantity. So a typical SLA printer will include the laser as the light resource and the mirror to control the direction of the light, the printing head to hold the printed object, and the resin as the materials. So the mirror can change the direction of the light to control the spot in which place you want the material to be crossing onto the printing head. So it's just like you use a laser and the pen to draw the lines on each layer from different shapes. Then you stack all of the different layers together to form the 3D object. So the main working mechanism is like shown this figure. Then when you print the first layer, the printing head will immerse into the resin until the bottom, until there's only one layer of thickness between the bottom and the printing head. Then this layer of resin will be cross-linked onto the printing head. Then the printing head will lift up to let the resin to flow back into let the resin to flow back into the central area. Then the printing head will immerse again into the resin to leave one layer of thickness. Then the second layer will cross-link onto the first layer. So after many cycles, the whole object can be printed. So please remember that we need this resin flowing back process to enable the every layer be cross-linked onto plant form. This will contribute to the most of the 3D printing time. So SLA has been widely used in biomedical research, such as this biodegradable tissue engineering scaffold. So the living cells can be grown inside this tiny structure with high viability. And SLA can also be used to print the hydrogel-based tablets and polybills with different shapes and different geometry and different drug components. So the second technique based on light is called digital light processing, DLP. Actually, DLP is the second generation of SLA. The only difference is that they introduced a digital micromirror device that can convert the laser beam into the light with different shapes. So it means for SLA, you just use a laser pen to draw different lines from one layer, but for DLP, you use the light as a stamp. Then you form a one layer and once. So in principle, the DLP should be faster than the SLA, right? Because the exposure time should be less. But actually for DLP, you also have this resin flow bank process. So actually the whole printing time is still not so significantly different. Because it's a simplified process of the projection light, DLP has become the most widely used photopolarization-based 3D printing method in both the industry and the scientific research. So for example, this work, they use the biodegradable materials to be printed with DLP to form this biodegradable and bioactive scaffolds that can enable living cells growing size and with additional functionalities. And DLP has also been successfully used to print the multivascular networks and the functional intravascular topologies. So as shown in this figure, this complex structure can mimic the ventilation and oxygenation that's generated by this human lung structure. So this is a very advanced step toward the real 3D printed human organ. And the next technique I want to introduce is it's called continuous liquid interface production, so-called CLIP. For this technique, the major difference is that they introduced an oxygen permeable window at the bottom of the resin container. So the oxygen can permeate through this window to the liquid resin and create a dead zone of at the bottom of the liquid resin. So in this case, it's not necessary to let the printing head lift up and let the liquid resin to go back to the central area for cross-link of each layer. The printing can be a continuous process. So this resin flow bank process is not necessary anymore. Then this technique can save a lot of time for the printing. And also the printing resolution can be greatly improved. So CLIP has also been used for the biomedical research, such as these drug delivery devices. They can design different geometries of the device and control the drug release kinetics. Another very good example is that they can use the CLIP to print this drug absorber implant. So, and we know that any cancer drug is very toxic because the tumor cannot absorb all of the anti-cancer drugs administered to the human body. So these drugs will go to the health tissue after the system circulation, then cause very strong side effects. But they implant this absorber just after the tumor along with the blood flow direction. Then the drug molecules not absorbed by the tumor can be captured by the absorber. So this can greatly reduce the cytotoxicity of the drugs to the human body. So I think this is a very interesting example of the application of CLIP. And also CLIP can be used to produce these microneedles with the micrometer scales, because the needles is very small and tiny, so they can greatly reduce the pain caused to the patient upon the drug injection or vaccinations. So these microneedles are very promising in the future to replace the current needles. So the most recent photopolarization-based 3D printing is called volumetric 3D printing. This technique completely abandoned the previous layer-by-layer lithography method. Instead, they use a tomographic reconstruction method. So the container for the resin will be rotating on this DLP project light irradiation, and only the specific area of the resin will be cross-linked under this light irradiation, and the 3D object will be formed. Very important thing is that the whole printing can be finished in less than 1 minute. So this is very fast compared to all of the previous 3D printing methods. And the resolution is almost the same with previous SLA-based 3D printings. This could be very useful for the bioprinting, because in bioprinting, if they use this technique, they have very short light irradiation time. So the cells grow inside the living tissue constructs can maintain very high viability. So these techniques, I believe, is very promising. Actually, this new 3D printing method is independently invented by a group in US and also another group from EPFL. So the last technique I want to introduce is the two-photon micro SLA or two-photon polymerization. So the difference of this technique with all the other photopolymerization-based 3D printing is that they use a different light resource. So they use a light called the femtosecond laser. For this laser, when the materials was irradiated, only the focus point in the materials will be cross-linked. In this focus point area, the materials will get solidified. But because of this focus point is very tiny, so they can make use to produce very tiny objects with high resolution. Just like this little church produced by two-photon polymerization, the whole size is less than 100 micrometers. But because they use a very special light resource, so the instrument is actually very expensive. And also you can see it's a very big machine. So their application in biomedical application or biomedical research are still quite limited. And also because they can only generate the very tiny objects, so the printing speed is not so fast compared to the conventional light-based 3D printing. But there are indeed some applications for the construct the 3D microenvironment with living cells. Like this work performed by my colleague at the ETH, Dr. Qin Xiaohua. They 3D print a lot of different projects that with living cells growing inside, and they can use the near-infrared light to control the cell growth behavior in the living tissues. So this could be very useful for the future tissue engineering research. Because there are actually a lot of new techniques based on photopolymerization 3D printing developed recently. Due to the limited time, I can't introduce all of them. So I will conclude here. Then, from all the previous introduction, we may know light-based 3D printing or the photopolymerization-based 3D printing has very high resolution and service quality. So they have very good potential in the personalized medicine. But currently, we still have very limited biocompatible and biodegradable materials can be used. In the same time, the new techniques are coming or are being developed. So that's a large space to explore the potential applications of these techniques. I would say I believe the 3D printing, no matter based on light or extrusion, will achieve significant progress in the clinical applications in the near future. Like, for example, the 3D printed human organ. But this would need the great effort from the scientists from all different areas, from cancer to materials to pharmaceutical science to biomedicine. So this won't be an easy work, but I think it's very promising. So with this, I want to thank the introduction from SwissNext and also the support from ETH, the help from Chinese Science Club in Zurich. And thank you very much for your attention to the audience. So my colleague Nevna will introduce more details about the DLP and what we are doing now. Thank you very much.
Okay, thank you, Dr. Bao, for the interesting talk. Now I'm delighted to introduce our second speaker, Nivina Paunovic. She is PhD student in the Drug Formulation and Delivery Group at ETH Zurich, working on the project of biodegradable materials for 3D printing of medical devices. She is a licensed pharmacist, holds a master in pharmaceutical sciences from University of Belgrade, and a master in drug sciences from University of Basel. So, Ms. Paunovic, the floor is yours. Okay. Sorry, Nivina, we cannot hear you. Sorry, we cannot hear you. We cannot hear you.
Because of this. Can you hear me now?
Yeah.
Yes, okay. You can just, yeah. Okay, sorry for this. Okay, so let's then come back and start from the beginning. Okay. So again, thank you, Living, for your kind introduction. And yes, so I will tell you something more now about 3D printing of personalized medical devices with focus on bioresorbable medical devices. So first, I wanted to start with the concept of personalized medicine. And as you probably know, the traditional medicine is based on the concept one size fits all. That means that all the patients should receive the same therapy or the same treatment. But if we would perform certain kind of tests, including laboratory analysis of blood, urine, or tissue analysis, or analyzing the DNA of patients, we would realize that they are quite different, that they have they express different genes, maybe more or less, that they have a different different amount of certain proteins or actually enzymes which will metabolize the drugs, and this will impact highly how the drugs are acting in the human body. So are they efficient, and what are the side effects? And if we would perform this kind of test, then we could divide the patients in certain groups, and we would decide which group should receive which kind of treatment. But we can apply actually the same concept with medical devices if we would use medical imaging. Because if we would use CT scans or MRI, we could also obtain personalized information about anatomy of an individual patient. So we could get information about complex geometries of individual patients and create medical devices just for that one patient. And first of all, I would like to start with the surgical models. As Dr. Bao already mentioned, they are very useful. They are not something that we consider as medical devices in a traditional way, so you will not insert them in the human body. But this can actually highly improve surgical outcomes, because surgeons can 3D print models of a very complex medical problems. So like in this case, you can see on this small picture, like in white, you're actually seeing a real model of a 5-month-old infant with complex cardiovascular obstruction. So it's really tiny, and the patient, and if a surgeon would start the surgery without creating the model, he would see for the first time some specific anatomical differences between this patient and the other patients he faced before. So they enlarged this model, created a big one, so they can anticipate all the mistakes and go through the surgery before the surgery actually happens. And it's also very good because you can train new medical professionals on these kind of models without any risk for the patient. Also, one of the surgeons from Yale University is also using these models, also of course to practice for the surgery, but also to get the informed consent from his patients and parents of his patients, because it's much easier to explain what will happen if you have a 3D model that can on which actually you can show what's happening during the surgery and explain it to a non-medical patient. So without medical background, I mean. This brings me to personalized medicine, like how we imagine personalized medical devices. And I would like to start with mouth guards, because I think this is something that it's very easy for everyone to imagine. We all have different maxillary anatomy, as you can see also on the pictures down. And if you need to wear a mouth guard, you want it to fit perfectly. So you want it to be really made just for you. And you do not want to feel any kind of discomfort, and you want certain drugs to be released you want controlled release, you want localized drug delivery. And if you go to a dentist, then they have this kind of scanners. So they are quite small and very convenient. So there is no need for anesthesia or whatever. We will get some kind of like CT scans that we can further use to create this maxillary anatomy model. And then based on this model, we are creating a mouth guard that should perfectly fit the anatomy of an individual patient. Of course, the challenge is, as always, how we develop materials, because you want material that will be comfortable enough. You also do not want something, in this case, you actually do not want something that will be biodegradable. You want it to release the drug in a predictable way. So you need to think about how your drug will be released from this material. And with 3D printing, you can actually achieve spatial control. So you can decide that you want to treat just the left or the right side of the oral cavity. You can also decide that you just want that the drug should be in contact with gum or just with contact with teeth. So you have plenty of options which would not be so easily achieved with traditional manufacturing methods. And if you have this spatial control, this gives you localized drug delivery, which means boosting efficacy of the drug and causing less side effects. Plus, you get something that is personalized, so it's very comfortable to wear. Also, people are now developing biodegradable mouth guards just to show that they are sustainable, but this is another story. For now, I would say that the biggest potential that 3D printing of bioresorbable medical devices showed is actually in making bioresorbable tracheal splints. As Dr. Bao showed you shortly, this is actually very important for a disease, or actually disorder, called the tracheal bronchomalacia. It's not so rare. Actually, 1 in 2,000 babies will be affected by this disorder. And this means that, so between your throat and the lungs, there is a system that allows the air to go this way. So if there is a weakness somewhere in between, the respiratory tract in a way collapses and patient stops breathing. In most of the kids, so for most of the kids, they will grow out of this condition. So this will be, the tissue will grow on its own, it will become strong enough, and just after 2 to 3 years, they will be able to breathe on their own without any episodes of breathlessness. But in 10% of patients, this can be very severe, and these patients end up on a daily basis, at least on a weekly basis, being reanimated because they stop breathing. And we are now talking really about babies, about infants, about like 2 to 3 months old who cannot breathe on a weekly level. And this is what happened to this first patient who got bioresorbable tracheal splint made by 3D printing. And I need to tell you that this is not a common procedure. So you will not get an implant made by 3D printing so easily, although maybe it looks safe and maybe extensive studies are conducted already, but the problem is that you need the regulatory approval. And this happens just in these like life-threatening situations when the risks are lower than potential benefits. So in this case, FDA decided that this condition deserves a chance to try 3D printed tracheal splints made by bioresorbable polymers. And what they did, so they obtained CT scans, then they created a model, and then they created this personalized tracheal splint that can provide support in this weak area. And then they sew it, and the patient started breathing immediately. So this tracheal splint should also be elastic, flexible enough to allow like the movements of the respiratory tract, and also to allow tissue of the patient to grow around. What is a nice thing with this is actually that, of course, the patient should, as I said, after 2 to 3 years, they should already be able to build their own tissue, and this biodegradable splint will degrade after up to 3 years, they say, and then the patient should be able to breathe on its own, which actually happened in the case of little Feiba. So After this, I would like to tell you something more about what we are doing here at ETH. So we are trying to make personalized bioresorbable airway stents by 3D printing. Why we need airway stents? If there is a disorder called central airway obstruction, which actually means that there is some kind of obstruction in the upper part of the airways, and patients are then affected by severe breathlessness. Their quality of life is impaired, and they need immediate relief, which can be achieved by airway stents. And they are actually tubular medical devices that can easily open airway stent, that can easily open the obstructed part of the airways, and they should actually perfectly fit anatomical needs of the patient. And here is where the problems come. So at the market currently, we have silicone stents and metallic stents. Silicone stents are good because they are biocompatible and they come in limited number of sizes and shapes, but for most of the patients, it's possible still to fit somehow to their needs with these limited numbers of sizes and shapes. But the problem is that when you have a more complex anatomical changes due to obstruction, it's also a problem where you have older patients, or if you have very young patients who will grow. If they grow, their airways are also becoming bigger, and then these stents can move. And if they move, there is a need for an additional surgery to remove these stents because they should not move in the respiratory tract, and they will not disappear, so they will stay there for forever. And for metallic stents, they are actually considered as permanent ones because they are quite strong. They open, but then they get embedded in the tissue, and it's really hard to extract them after. So even if you would extract them, you would actually induce such a high injury that you are even at higher risk to get obstruction in the upper part of the airway. So ideal stents should be personalized. That would that would actually decrease the risk of the migration. It should also be biocompatible as every medical device. It should be biodegradable because, as I said, if something happens, and also like after this medical device fulfills its role, it should disappear. Or if it moves, it should also be able to disappear after a while. And these materials should also be thin and flexible. These stents should be thin and flexible, and we should also be able to produce them fast and ideally with a low price. So where is the 3D printing in this story? We, of course, want to make personalized medical devices which are less prone to migration. But this is something that is happening now for decades. So surgeons are doing what they had at the moment. So they were actually cutting different silicone stents, combining them, gluing them. And of course, you can imagine that this did not ever get approval from regulatory bodies, but still it helped patients. And we came with this to the point where we can create personalized molds for silicone stents. So you can create personalized silicone stents, but the problem is that the patient who cannot breathe now needs to wait for 2 weeks for these molds to be produced and then to get a customized stent. And as you can imagine, this is not so convenient if the patient cannot breathe. And this is why we would like to introduce digital light processing in this field, as these printers have a desktop size, so we can easily put them in a hospital setting. As Dr. Bao showed you, 3D printed objects have a really high resolution, high surface quality. Printing is very fast. And the good thing is also that the printing, like the time of printing actually depends on the height of the object. And what is achieved so far is that people are doing customized airway stents. So, but they are usually made by FDM printing. So the resolution is not so high. They are not biodegradable, and their mechanical properties are not ideal. And as you can see here on the other picture, also by FDM you can print molds that can then be used to inject silicone inside and to get personalized silicone stents. And this is something that is now approved also by FDA. Still, this requires certain kind of expertise and space where you can produce this kind of stents. So it's not so convenient to do it in a hospital. Why digital light processing? So as you probably now remember, we have a liquid resin from which we are getting solid object upon laser exposure, because inside the molecules are cross-linking with each other, creating a network, which is then creating a solid object. And with this, we are getting really high resolution and really high surface quality. which is the highest advantage of light-based techniques among the others. But still, of course, nothing comes without some challenges, and this technique also comes with a certain price. And this is that we need to develop these low viscosity resins to be able to get this high resolution and surface quality. And also there is a lack of biocompatible and biodegradable materials upon 3D printing. So even when we satisfy these requirements, as you can see here, so we can get an elastic medical device, but then this one will be probably too weak to resist the forces that, for example, we need to have, that we are actually having for airway stands. Because when you develop an airway stand, you want something that is elastic enough to be folded and easily inserted into patients. You also want something that will mimic human anatomy, so something that will easily move with the patient, allow them to breathe and being quite flexible. But you also want something that is strong to open these obstructed airways. And if you achieve one of these, you usually cannot combine them. So if they are elastic, usually they are brittle, but they are weak. But if you get something that is quite strong, then it's usually brittle. So regarding the opportunities of 3D printing, I would say that definitely the highest opportunities are in personalized medicine. Regarding the drug formulations, as a pharmacist, I would say that this will not that 3D printing will not replace traditional ways of manufacturing drugs, because as Dr. Ba also showed you, there is always a certain threshold where it becomes financially acceptable to use 3D printing. And usually for traditional dosage forms, we do not have this kind of problems because we can make them more or less personalized. There are some special cases, and I can imagine in a pharmacy one day having a 3D printer and coming with your lab results and using different types of vitamins in one tablet that will have different taste or being in different shapes for kids or something like this. But the biggest potential, like from this technique, should actually be in manufacturing medical devices, personalized medical devices, where we can get high complexity at lower price. So we are not limited by the manufacturing method. We are not limited by the time. We can produce these medical devices in a short period of time. We can design them in any shape we want. And it should be quite affordable when we compare it, how much it would cost with traditional manufacturing methods. Also, I would like you not to forget that models for surgeries are also very important, and that we can with this anticipate mistakes, and that we can train medical professionals without risks for patients. And also informed consent is really important for patients and their parents, and this would allow us to explain them better what's happening during the surgery. But I would like to finish this talk actually with challenges and what should be done in this field, because the biggest problem, the biggest challenge is lack of suitable materials. And this is what we need to work on if we want 3D printing really to be implemented in medical practice. Also, the challenge is in massive production, because the price will always be a question. And then we come to regulatory approval, which is very complex if you start using new manufacturing methods with new materials, and then you are creating some objects, and who will guarantee that these objects are really what you wanted to design? There are many safety concerns. Are your medical devices releasing some toxic or harmful substances? Are they, how stable they are, for how long? Can a medical doctor operate with this kind of 3D printers? And then it also comes the question about responsibility. Who is responsible if this kind of medical devices cause any kind of harm? What if they break? What if something is wrong? Who is responsible for that? And this is why we need to work, as Dr. Ba also said, like we need to have teams of professionals in different areas and try to solve these problems and help our patients to benefit from this amazing technique. And with this, I would like to acknowledge our group of drug formulation and delivery, and also our collaborators in complex materials at ETH. Also, I would like to thank our collaborators at University of Zurich and University Hospital, and of course, the Swiss National Science Foundation for funding our research. And with this, thank you all for your attention, and I'm looking forward to your questions. I cannot hear.
Okay, thank you, Ms. Paunovic, for the informative presentation. Now let's come to the Q&A session. So we have some questions from the audience. So the first one is from Xiaoyu Du. Thanks for the great introduction. I would like to ask if 3D printing can print some closed form scaffold. Maybe to Dr. Bao. Can you please unmute yourself? Can you please unmute yourself? Yeah, okay.
Can you hear me now?
Yes, yes.
Okay, could you repeat the question? I didn't hear it clearly.
Oh, sorry. So it's from Xiaoyu Du. Thanks for the great introduction. I would like to know if 3D printing can print some closed foam scaffolds.
Closed form?
Closed foam.
Closed form.
Yeah, sorry.
So for closed form scaffold, it's indeed a challenging for the conventional 3D printing, because normally for light-based 3D printing, you need to remove the residue materials from the 3D printed object. If you have a closed form, then maybe the inner part of the object will still contain the unreacted materials. Then you can't remove these materials from the object. This will cause problems. So normally you should have at least the tiny channels to connect with the outer and the inner structure. Then you can try to clean the objects to remove the unreacted materials or the residue monomers. So it's indeed a challenging.
Okay, thanks. Then and the second one is if you are producing biodegradable materials, you would need a trigger that controls when the degradation should start. Otherwise, degradation might begin too early or too late. Do you work on such issues?
Do you want to come?
Mute, but maybe I can reply.
Yeah, you can reply.
Okay. Any.
Sorry, we cannot hear you.
Cannot.
Now we can hear you.
Okay, great. Yes, of course. So any biodegradable material, if you do not protect it somehow, it will start degrading immediately. I mean, not every, but most of these we are working on currently because they are based on polyesters. And then it's just the question in which moment you are losing mechanical properties that you need for a certain application. Because everything will start, if it's biodegradable, it will start degrading. Some of them faster, some of them slower, but you cannot stop it. So it's just, you can sometimes do some kind of coating or protect certain parts and try to slow down the degradation process. But there are also many different ways how you can stop this. You can also combine different functional groups that maybe will be cleaved slower. But the problem is that, I mean, it's not a problem, it's a good thing in a way, because the degradation starts. It depends in which moment you are losing the properties you want to have for a certain application. But for sure, there is no object that will provide this mechanical support for, let's say, 3 years, and then in 3 years it disappears. So it will start gradually to lose its mechanical properties.
Okay. So Dr. Bao, do you want to add something to this? Dr. Bao, do you want to add something to the question?
No, we can't hear.
You not hear.
Can you hear me now?
Yeah.
I was asking if you would like to add something regarding this question.
For the responsive materials, actually, we can also add some triggers in addition to the degradable materials. We can induce some stimulus responsive linkers in these biodegradable materials. Then with additional functions, when you apply this material in the specific areas with different like different pH or different enzymes, then maybe you can accelerate this degradable process for spatial purpose. But for the conventional degradable materials, we want to introduce this kind of functionality.
Okay, thank you. Then another question from Shai Xue. Thanks a lot for your impressive presentation. I would like to ask what kind of bioabsorbable materials are you using currently? And do you think SLA or DLP can be used for cell printing? If yes, how does the laser effect on cells?
Yeah, maybe I can answer these questions. So the biodegradable materials we are using now is actually it's not very special materials. We just screening the previous biodegradable polymeric materials normally used for the biodegradable elastomers. Then we'll combine the most interesting ones for our 3D printing and test their mechanical properties, degradation properties, and in vitro and in vivo performance. Then we get to the final optimized materials. So this there will be a lot of pre-screening work and the optimization work to get the best result for specific applications. So I would say that the main polymers or the main 3D printing materials that we are working on are based on the polyester structures. And for the cell printing, of course, it's possible because actually, as shown in my slides, a lot of work has been done with the 3D printed structure with cell lengthened constructs with living cells inside. And just I show this volumetric 3D printing with very short print time, you can already get the whole structure. So this is very useful for the bioprinting. So the living cells inside the structure won't be affected at all by the UV light. And even for the conventional SLA or DLP, the damage of the UV light or the laser irradiation to the cells are quite limited. After compare the viability of the cells by the in vitro experiment, you can see the cells can still grow very well inside the tissue engineering scaffold. And also, what I want to say, another point is that okay, so maybe when I realize it, I will talk again about this. Yeah.
Okay, thank you. So I would also like to ask to Dr. Bao. Earlier, you showed us a few applications of 3D printing in the fight against COVID-19. So in the very beginning of your presentation. So I would like to know, are these applications commonly used and accessible in Switzerland? And how about in China?
And for this 3D printing facility to fight against the COVID, actually, that's only for use in emergency situation like this very fast spread of the coronavirus. So when the medical resource is very occupied by this COVID patient, this 3D printing may play some roles inside. But when the situation is released, the urgency is reduced, you don't need this kind of facility anymore. But for conventional like the printing of the surgical models, I think most of the large hospitals will have this kind of special 3D printing facility to fulfill the requirement from the doctors, the physicians for the surgery. And not only in Switzerland, but also in China. So I think this is won't be no big difference for the facility itself. Because China also has a very strong manufacturing ability to build this kind of 3D printers for all kinds of 3D printers you can find in China. So I don't think that's a big difference. And also the facility should be available for most of the large hospitals.
Okay, thank you. The next question to Nivina. Based on the current knowledge, are there any adverse events for patients receiving transplantation of bioresorbable airway stents?
So there are no 3D printed airway stents, so you can. Thanks. So there are no 3D printed bioresorbable airway stents. but there are bioresorbable air stents made by other techniques, and they formed some small pieces. And so these pieces were really brittle. So we are trying to develop elastomers, and these materials are quite stiff. They are more competing with metallic stents. And these solid pieces can be embedded into tissue and actually create some injuries. And so these are the potential risks, I would say, with currently available bioresorbable stents. So we would like to change this so they degrade in a form of hydrogen.
Okay. Okay, then maybe last question to both of our speakers. I think you already mentioned something during your presentations. So what are the main challenges that you foresee in terms of universalizing personalized medicine produced by the light-based 3D printing?
So this is for me?
Doctor. Yeah, to both of you, yeah.
Okay, thank you. Because at the beginning I couldn't hear it. But there's double sounds inside.
Sorry. Shall I repeat the question?
Yeah, maybe it's better to repeat. Yeah.
Yeah. So, what are the main challenges that you foresee in terms of universalizing personalized medicine produced by light-based 3D printing?
Yeah. Thank you for the question. Actually, the light-based 3D printing is quite new or quite special compared with the conventional 3D printing based on extrusion, because you need to use the specialized materials. So for this approval of the material to be used for medical device or other 3D printed devices, this will be much more complicated to be compared to the conventional 3D printing. So how to prepare the biocompatible and biodegradable materials can be used for light-based 3D printing and also fulfill the requirement for the personalized medicine. There's a balance you need to find it. So this will be the major challenging how to do it. You need the very good properties to support the application, and also the very safe property to make sure there's no harm for the human body. So you need to find a balance. This will be the most challenging thing. So the material development will be, I think, a very important step for further apply this technique in the clinical or biomedical applications.
Okay, thank you.
Yeah, I just remember the one additional point for this previous question raised by Xue Shuai. So actually now there are already new light-based 3D printing system using the green light, red light, or near-infrared light invented. So conventional light-based 3D printing, we use the UV light. So it's we won't say it's not harmful for cells, but if we use the red light or near-infrared light, of course it's will be we can say it's safe, right? So this we just read the recent papers. I think it's published just this year. So I think in the future the technique will be much better with the new adventures. Thanks.
Okay. So, well, due to time constraints, we will have to end the Q&A session. So thank you very much to Dr. Bao and to Ms. Paunovic for your available time and your deep insights. So to our audience, if you have more questions, please send them to the email address on this slide. We will forward your questions to our speakers and give their feedback to you. You are also more than welcome to scan the QR code to follow us on our social media channel and subscribe to our newsletter. Thank you indeed for your active participation, and looking forward to meeting you in our next events. Thank you very much, Dr. Bao, and thank you, Ms. Paunovic. Thank you.
Thank you very much.
Thank you.
Bye-bye.
Bye.